Hello everyone, welcome to the ITLS Academy. Today we are going to discuss another topic, that is your estimation of cholesterol by Zerk's method. Okay, so we are going to discuss about the estimation of cholesterol by Zerk's method. So let's start the lecture. Aim to estimate the amount of cholesterol in the given food or serum sample. Principle: cholesterol is a steroid lipid. Amphipathic in nature. Amphipathic in nature. It consists of basic cyclopenton per hydrophenol 3 nucleus. It is synthesized in liver, from acetyl coenzyme. It acts as a precursor for steroid hormones and vitamin D. Then we talk about the Zerk's method. In Zerk's method, the protein which are present into the sample which are present into the sample are precipitate when ferric chloride and CH3COOH reagent is added. Then after that, we filter the protein, filter the protein with the help of the concentrated sulfuric acid, and in the presence of excess sulfuric acid, in the presence of excess concentrated sulfuric acid, cholesterol present in a sample is dehydrated. Cholesterol present, in the sample is dehydrated. Cholesterol which are present into the sample is dehydrated. Suppose that this is your cholesterol. Now this cholesterol is dehydrated. When in the presence of excess concentrated sulfuric acid, then it is converted into the cholesta 3,5 diene. Then it can be catalyzed by the ion, that is ferric ion, and a red color complex is formed. Red colored complex is formed. Then the intensity of the red color we measure. Intensity of the red color is measured at 560 nanometer. Okay, now see the structure of the cholesterol. Here a ring like structure, you see that? Just a minute. Like this. Then here, just a minute. Here, like this. Then this. Then here CH group is present, which is attached to the CH2. Then here it is attached to the CH2, CH2, CH, CH3, CH3. Then this CH is attached to that CH3. Then here CH3 group is present. Here CH3 group is present. Here OH group is present. And this structure is known as a structure of cholesterol. Structure of cholesterol. Okay, now come to the next slide, that is your reagent. Reagent preparation. So first reagent that we required is a stock solution. So what we have to do, we take 100 milligram of cholesterol and it will be dissolved into the 100 ml of glacial acetic acid. Just simply take 100 milligram of cholesterol and dissolve into the 100 ml of the glacial acetic acid. With the help of this stock, you have to prepare the working solution. So for the preparation of the working solution, what we have to do, we take 4 ml of stock and makeup volume 100 ml with ferric chloride, ferric chloride, acetic acid. Okay, makeup volume means that if we have this amount 4 ml of the stock, then we have to add ferric chloride acetic acid 96 ml. This is known as the makeup volume 100 ml. Then we required ferric chloride 0.05% in acetic acid, which means that we take 0.05 gram of ferric chloride plus 100 ml of acetic acid. Simple. Then we required some another reagent also, that is your concentrated sulfuric acid, glacier acetic acid, and preparation of the unknown solution. Unknown solution like sample. So for this, what you have to do, you have to take a 20 ml of the sample plus 40 ml of chloroform and centrifuge. Then take the supernatant for further experiment. So this is how we prepare the sample. Now come to the procedure. Now procedure. So for procedure, we required 7 test tubes. 1, 2, 3, 4, 5, 6, 7. 5, 6, 7. Now what we have to do, we have to add the working solution of the cholesterol into each test tube, and it is range in between the 0.5 ml to 2.5 ml. In a first test tube, add 0.5 ml, then 1 ml, then 1.5 ml, then 2 ml, then 2.5 ml. Then in a last 2 test tube, you have to add the sample, 1 ml sample in a first one, 1 ml sample and 0.5 ml sample. Then you have to make up volume 5 ml with ferric chloride. Means that in a first test tube you have to add 4.5 ml ferric chloride, then 4 ml ferric chloride, then 3.5, 3 ml, then 2.5. Okay, in a 6 test tube you have to add 4 ml and 4.5 ml in a last test tube. Then after that you have to add 3 ml concentrated sulfuric acid into each test tube. Add 3 ml concentrated sulfuric acid into each test tube. After that, keep it at room temperature. Keep it at room temperature for 15 minutes. Then take a absorbance at 540 nanometer. Okay, then draw a graph between the absorbance and concentration of cholesterol. You get the straight line, and the concentration of the samples are lie in between this. By this, you have to calculate the result. So this is how we perform the estimation of the cholesterol in a given food sample. I hope you will understand, but if you have any query related to this, you are free to ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also ask your query to the helpline number that are already mentioned in your slide, that is 7080833450, and visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.